Modern cells are, of course, very different from what the most primitive cellular life would have looked like. Let's take a couple of minutes and talk about some of the features that we find in modern cells. Remember, all cells require energy. They require a continuing source of energy. Cells will use that energy to do work. Remember that there is a particular molecule inside of all living cells that serves as sort of a cellular currency, and that molecule is ATP, or adenosine triphosphate. Adenosine triphosphate is produced during a couple of different types of metabolic reactions. Cellular respiration and oxidative phosphorylation is a very common way for cells to produce their ATP. Now, prokaryotic organisms are also capable of cellular respiration and oxidative phosphorylation, but prokaryotic cells, some of them at least, can also produce ATP through a process called fermentation. So there are different ways for different types of cells to produce their ATP. But all cells use their ATP similarly. They use ATP for a process known as energetic coupling. Essentially, ATP is used to capture energy that is released during catabolic metabolism, during the kinds of reactions where molecules are broken down. Covalent bonds are broken and electrons are released. Catabolic metabolism releases energy. It's referred to as being exergonic. And that energy that gets released would be lost to the cell if it wasn't harvested into ATP. The ATP molecule itself can then have its outer phosphate group broken off so that that stored energy, that potential energy that was harvested, can be used by anabolic metabolic reactions, the kinds of reactions that require energy to build molecules, what we refer to as endergonic reactions. All living cells use ATP for this energetic coupling between catabolic and anabolic reactions. I have a note here on the slide to remember that there is a second currency molecule inside of cells called GTP. GTP is not used nearly as often as ATP is, but Guanosine triphosphate is also present in cells. We will mention it a few times this semester as we learn about the inner workings of cells. GTP is likely the more ancient molecule here. In other words, ATP likely evolved from GTP. Now, we also need to review the way that cells get energy to produce ATP. And if you recall from your BIO312 class, your introductory class, cells make ATP by breaking down or oxidizing various materials, including glucose. So we can categorize organisms by how they get energy necessary to build ATP, to produce ATP. Organotrophs, or what are sometimes called heterotrophs, get their glucose by consuming organic compounds, sometimes consuming other organisms. They get the glucose by eating other organisms or other sources of organic compounds. That glucose, again, is oxidized. It is broken down so that the energy from the glucose can be harvested and can be used to produce ATP. Phototrophs, also called autotrophs, are the kinds of organisms that don't need to consume glucose because they can produce their own glucose. And they do that by harvesting the energy from sunlight. You can think of them as consuming light and producing or building their own glucose. They can then turn around and oxidize that glucose to make their ATP. The last category are the lithotrophs. This is a smaller group of organisms on our planet. These are the kinds of organisms that consume not glucose, not organic compounds, but inorganic materials. They can then break apart those inorganic molecules to obtain energy to produce ATP. We've said that all cells use DNA as their genetic material, and we should take a minute to remind ourselves of how the information in DNA, the genetic information, flows within a cell. How does that information get used within a cell? And that brings us, of course, to the central dogma of biology. The central dogma helps us understand how information flows from the DNA molecule into the traits that cells have. Remember that the central dogma has 3 distinct statements in it. The first says that DNA is transcribed to make a molecule called RNA. And that process is referred to as transcription. The second statement says that RNA is translated to produce protein. That process is called translation. And the third statement tells us that proteins are what create the traits in cells. Remember that DNA is also used for something very different in a cell. It's not always used for gene expression. Sometimes DNA simply has to be copied or replicated. Any time that a cell is preparing to divide and produce new cells, the DNA molecule has to be replicated using what we call semi-conservative replication. And that's what we're looking at in the diagram at the bottom of the slide. We're looking at the uses of DNA inside of a cell, the way that the information in the DNA molecule flows. DNA can be used to make more DNA. That's what the blue arrow is showing us, through the process called DNA replication or DNA synthesis. Other times, the cell uses its DNA to build proteins. That's what we refer to as gene expression. The genes inside the DNA molecule are transcribed to produce RNA. And then that RNA is translated to build proteins. And it's the proteins that then give the cell its traits. All cells contain that organism's entire DNA molecule, or its entire genome, its entire collection of genes. Now that makes sense if you think about a single-celled organism. It makes sense that if the organism is composed of one cell, the DNA molecule inside that cell must have all of that organism's genes. When we think about multi-celled organisms, however, it's sometimes tempting to think that different cell types have different types of DNA in them. That a liver cell, for example, must contain the portions of DNA that code for proteins that the liver cells need, or that neurons contain DNA that has genes in it that build proteins that neurons need. In reality, every cell inside that multi-celled organism has the entire DNA molecule in it. So that tells us something. That tells us that different cell types have the entire genome, but only express a portion of that genome. Different cell types express different sets of genes inside the DNA molecule. Now, regardless of whether you're looking at a single-celled organism or a multi-celled organism, there are some genes in the DNA molecule that get transcribed continually, 24 hours a day, 7 days a week. We call this type of gene expression constitutive expression. That's a miserable word to spell, but we pronounce it constitutive. You can imagine some of the genes that are constitutively expressed. Think, for example, about the gene that codes for the enzyme called RNA polymerase. Remember, RNA polymerase is the enzyme that does the work of transcription, of using DNA to make an RNA. RNA polymerase is required around the clock in a living cell because cells are constantly transcribing genes to build proteins. So a cell has an ongoing need for the RNA polymerase enzyme, and a cell will constitutively express the gene that makes that enzyme. There are a limited number of genes that are constitutively expressed. You'll sometimes hear those genes referred to as housekeeping genes, the kinds of genes that are needed around the clock. All other genes, in fact, most genes, are not constitutively expressed. They are expressed only when the cell has a need for that specific protein at that moment. These genes are under what we call regulated expression. You've probably heard people talk about genes being turned on and genes being turned off. Sometimes genes are not precisely turned off, but instead down expressed or down regulated. Other times, genes are turned on or turned up or up regulated in their expression. The mechanisms of regulated gene expression are very complex, as you might imagine. We're going to talk about a few of them later in the semester. All right, we're going to switch gears here and begin talking through a little review of biochemistry. Now, I know that the term biochemistry strikes fear into the hearts of many, but we will focus again on the parts of biochemistry that are important for us as students of cell biology. This should be review for you, but it's a good review because this information is important to us. On this first slide, I have listed some terms that you need to be familiar with. The word element and the word atom describe two different things, although we sometimes interchange them. An element is a substance that can't be broken down any further, and it cannot be converted into another element. Atoms are different. Atoms are the smallest particle of an element, so the smallest subunit of an element. Molecules, remember, are combinations of atoms. Atoms that are connected to each other through covalent bonding. And the other term we need to be familiar with is this term polymer. A polymer is a molecule that's built by combining the same molecule to itself over and over and over again. We refer to these subunit molecules as monomer units. When you join several monomers together with covalent bonds, you build a polymer. All of the individual elements are put together in what we call the periodic table of the elements. And you've seen images of the periodic table in your chemistry classes. Every element has a name and a symbol. You can see on this slide a designation for carbon with the symbol capital C. In the periodic table, we're also provided with two important numbers for this element called carbon: the atomic weight and the atomic number. Now, where these numbers will be placed in the box for a particular element, it varies depending on the version of the periodic table that you're looking at. But we need to have an understanding of what these numbers mean. The atomic number is simply the number of protons that you will find in the atomic nucleus for that element. So for carbon, the atomic number is 6. Over here on the right-hand side of the slide, you can see a drawing of a carbon atom. And there are 3 different particles that are represented inside the atom. There are electrons with a negative charge, protons with a positive charge, and neutrons that have no charge. They've drawn the protons and the neutrons as being large and the electrons as being small because, relatively speaking, protons and neutrons have most of the mass inside an atom. So at the center of the atom, you can see all of the protons and neutrons clustered together in what we call the atomic nucleus. The atomic number for carbon is 6. So we know that there are 6 protons in this atomic nucleus, and they're represented by the blue circle. The atomic weight for carbon is approximately 12. And since the atomic weight represents the number of protons and neutrons, we know that there must be 6 neutrons in this atomic nucleus, because 6 plus 6 equals 12. Now, neutral atoms, atoms that do not have a charge associated with them, have the number of protons equal to the number of electrons. Positive charge, negative charge. Charged atoms, on the other hand, have to have a difference between the number of protons and the number of electrons. And the charge will be determined based on which one is more abundant. If there are more protons than electrons, there'll be a positive charge on that atom. If there are more electrons than protons, there'll be a negative charge on that atom. A couple of other things that we need to be familiar with can also be found in the information in the periodic table. One of the things we use very frequently in biology in general, but also in cell biology, is this concept of molecular weight. All the molecular weight is the sum of those atomic weights of all of the atoms that are in a molecule. So, for example, for a water molecule, we would need to go to the periodic table and look at the atomic weight of both hydrogen and oxygen. We would take one oxygen atom and two hydrogen atom weights and combine them to determine the molecular weight of water. A couple other terms that we need to review are this term mole and this term molarity. When we talk about a mole of a substance, what we're talking about in practical terms is the molecular weight of that particular molecule in grams. So when a substance has a molecular weight of, say, X, then 1 mole of that substance is X grams. Molecular weight in grams. Remember from chemistry, a mole is actually describing 6.02 times 10 to the 23rd molecules of that substance. But again, in practical terms, a mole describes the molecular weight of some substance in grams. So for example, if we wanted to know what a mole of glucose is, we would take the molecular weight of glucose, which happens to be 180, and we would apply the label gram to it. So 1 mole of glucose is 180 grams of glucose. Knowing how to determine the moles of a substance is important because we often use solutions in the laboratory in cell biology. And in order to make up solutions, we need to be familiar with this concept of molarity. Molarity refers to the number of moles of a substance per liter of a solution. So if we go back to our example of glucose, a 1 molar solution of glucose would be 1 mole of glucose, or 180 grams, dissolved in 1 liter of solution. When we look at living cells, we can describe 4 critical elements. The most abundant, most common elements that you find are carbon, hydrogen, oxygen, and nitrogen. When you look at all of the matter that makes up a cell, 97% of it is composed of those 4 elements: carbon, hydrogen, oxygen, and nitrogen. Now, we compare those 4 elements to each other on what's referred to as the electronegativity scale, because it's going to tell us how those atoms tend to behave inside of a molecule. Now, the electronegativity scale is based upon the tendency of these elements to attract electrons to themselves. The highest electronegativity is found in oxygen, relative to the other 3 at least. Next in the group is nitrogen. And nitrogen is followed by both carbon and hydrogen. Now, carbon and hydrogen are slightly different in their electronegativities, but for our purposes, we're going to consider those 2 elements as having the same electronegativity. So carbon and hydrogen both have the same low electronegativity. And again, that means that they have a low tendency to attract electrons to themselves within a molecule. Now, why do we care about that? Why do we care about the electronegativity scale among those 4 elements? Well, it's going to help us understand how those elements are going to behave when they're combined into biological molecules. We need to take a step back here and think again about atomic structure. Remember, we said atoms are composed of protons, neutrons, and electrons, and every element has a different combination of those three types of particles. All of those electrons are buzzing around the atomic nucleus. They are in motion constantly around the atomic nucleus. We can predict, however, where an individual electron will be at any particular time around that atomic nucleus. Electrons, it turns out, are organized into these areas around the atomic nucleus, referred to as orbitals. And combinations of orbitals make up what we call shells around the nucleus. It's the very outer shell around an atom that is of interest to us in biology, what we call the valence shell. And that's because electrons within the valence shell are sometimes unpaired. And unpaired electrons are unstable. Unpaired electrons in the valence shell of an atom are going to seek out other electrons to pair with, because paired electrons are stable. Now, in order to achieve electron pairing, there are two ways to do it. Either electrons can be transferred from one atom to another, or a pair of electrons can be shared between two atoms. Take a look at the diagram on this slide. On the left-hand side, we see a drawing of two different atoms. And you can see the atomic nucleus in the center, and then this gray area, which represents that sort of cloud of electrons buzzing around the atomic nucleus. So there are different numbers of orbitals and different numbers of shells depending on what element you're talking about here. But out in the valence shell around both of these atoms, we can see a single unpaired electron drawn in red. These two atoms are unstable right now because they have unpaired electrons in their outermost or valence shell. One of the ways to solve this problem, to make both atoms stable, is for them to come together and share that pair of electrons. And that's what we see here. So this is a very simple molecule made up of two atoms, and those two atoms are sharing a pair of electrons. This is what we refer to as a covalent bond. All a covalent bond is a pair of shared electrons between two atoms. Now next door on the right-hand side of the slide, you can see an example of two atoms, again with unpaired electrons in their valence shells. But instead of coming together and sharing this pair of electrons, one of the atoms is going to donate its unpaired electron to the other atom. So now this atom has lost an electron, and this atom has gained an electron. They're still coming together, but they are not joined with a covalent bond. They're joined instead with an ionic bond. This atom on the left has lost an electron. It donated it over to this side. So this atom now has one less electron and a positive charge. This atom now has an extra electron. It has gained an electron, so it now has a negative charge. Any charged atom is referred to as an ion. This is a positive ion, this is a negative ion, and when they come together like this, they are held with an ionic bond.